I'll try to sort of continue this line of work on modeling evolutionary dynamics in cancer research. And this is the place I work, is the Moffitt Cancer Center. So I'm a group leader at the Integrated Mathematical Oncology Department here at the Moffitt Cancer Center in Tampa, Florida, sunny Tampa. Although I don't think I have anything to miss while I came here to Poland yesterday. Temperatures are about the same right now. I'll try to be as concise as possible, but I usually when I get excited, and today I'm getting very excited, this is a great place to be for me. I tend to talk faster, so I'm hoping that most of this presentation will come up in English. I can be confident about that, but you know, in case you want to contact me after this, trying to clarify what I said, this is a couple of ways you can approach me, and also I'd like to clarify that I'm happy to share all the information, so you want to take pictures or... I know that people have been doing that already, but obviously I'm more than happy for the information to come out any way possible. So also because the papers have been published mostly in journals that you probably don't read very often. But anyway, information out there. Now, because I didn't know when I prepared this presentation that I wouldn't be the only cancer researcher, I decided to come with a few slides in which I introduce a bit where we are right now and what we actually need to come with different approaches to treat cancer. And obviously cancer is a very, very old disease, and we are quite lucky of having been born in this time and age in which we have different approaches to treat cancer. For most of the history of mankind, treating cancer was possible, a very, very painful process. It's only relatively recently that we found out that we can poison tumor cells faster than we can poison ourselves, and use that to our advantage. And use things like radiation therapy, where the idea is that you can actually poison parts of your body as opposed to your entire body in the process of trying to kill cancer. Targeted therapies are basically the result of people putting billions of dollars and lots of research in molecular biology with the hope that if you find one single mutation that drives cancer, you can basically kill the cancer, not taking into account the fact that cancer is a very complex process. That's something that the NCI and other institutions are beginning to recognize, although that's not necessarily something you can see if you actually check the website, where cancer is basically explained as an accumulation of mutations in tumor cells, ignoring the fact that actually is an evolutionary process driven by tumor heterogeneity, something that we only started looking at in the last 5 years as a community. Most of us were actually working, some of us were actually looking at it well before that, but most of us were ignoring completely the fact that cancer is this heterogeneous evolutionary process. Now, you look at this is something that I think sort of resembles a bit more the sort of perspective that a lot of people have these days when it comes about cancer evolution. Heterogeneity, phylogenetic trees, all these are important things, but I'm going to steal this slide from one of the previous speakers. Actually, this is a really old classic example of what happens when you actually exclusively center on the genetics of the problem, which is that obviously you're a cancer biologist and you look at this, you say, well, you know, I used to find a lot of the white ones, these days I find a lot of the dark ones. What is happening? And you know what's happening is the context. And if you just look at things in isolation, you might basically miss the context in which mutations actually confer advantage and in which basically selection takes place. So the work that my group has been working on the last few years is trying to understand this context by producing an ecosystem of the tissue in which tumor actually comes in and sort of takes advantage of in order to grow faster. And this is complementary to the phylogenetic, genetic-driven view of cancer. It just requires us to think that the space and the stromal interactions, so the other cells that are not part of the tumor, that actually could hinder or promote tumor progression, are actually something that we need to consider in some of the models, at least to figure out what drives the selection process for increasingly more aggressive tumor phenotypes. So I'll just basically describe as briefly as I can one of the projects that my group has been involved in, and I'll try to butcher the job of this young gentleman over the top, which is Arturo Araujo, a mathematical modeler in my group, working with Leah Cook, which is an experimental bone biologist who is working with us together with her boss, Connor Lynch, which is the partner lab in our department. And a very brief explanation of the biology that I'm trying to capture with this model, it comes here. Basically, your bone gets remodeled very, very frequently basically in order to make sure that it remains as strong as healthy as possible. And I'm not trying, so you won't find much cancer here yet, so just follow with me for a second. Every time that a microfracture is found, just because you, for instance, go for a run, there's a process of bone remodeling that will take place. For instance, the stem cell here basically finds that there's something missing, basically divides asymmetrically, producing this, let's call, bone producing cell, which starts secreting factors, which recruits these other cells that come in, basically fuse into this sort of Pac-Man that starts resolving the bone, releasing even more factors that keep these other cells proliferating, but not working in producing bone. At some stage, the Pac-Man thing, osteoclast, dies, the bone producing cells come in and start doing their job of producing all this bone back to where it was, but just healthier. So this is the process that we're trying to capture. And we're trying to capture all this because it's incredibly important, it's very dynamic, it's orchestrated in a very bottom-up way. And this is the place in which tumor cells will come in and try to disrupt the process. So we use an agent-based model in which the different cell types we found relevant to this particular process are included into these flowcharts, and where the physical microenvironment in terms of the signals, in terms of the bone, is basically described as these partial differential equations, which are then basically discretized in a grid. We have a lot of parameters which we scan through literature, but the important thing of working in a place like ours is that we have lots of willing experimentalists. I described Leah's work before, who have basically produced for us the characterization of the impact of the tumor cells when they arrive to the bone, which we can actually input into the mathematical model. Now, this is basically how this mathematical model looks like in terms of the domain in which the cells will actually come in and do the thing. And this is just one simulation. You can see all the, you see? Yeah. So, just trying to see if I can click on this simulation. Unfortunately, apparently the movies didn't come in. But basically, if you saw this, you'll see that all the cells, basically the cells that form part of the bone modeling unit, will come in. They'll start basically proliferating at a rate that is basically described by these flowcharts you saw before. They come in, they recruit other cells that come and destroy the bone. Basically, the whole process that you saw before in terms of these beautiful pictures that are actually created is recreated here as well. Now, the interesting thing about this is that as opposed to the one, this is one that emerges from the interactions at the local level of the different cells, plus the interactions at the non-local level through the secretion of molecules. All these things are completely well studied when it comes to bone remodeling, when it comes to bone homeostasis. The next movie that I have here, basically, is the one that captures what happens when we actually have a tumor cell. You won't see it, but there is a small change at the beginning. It looks very, very subtle. The bone remodeling usually just looks like any other bone remodeling, but the tumor cell starts secreting a factor that basically disrupts the whole process, and it starts creating this increased proliferation of tumor cells, which recruits even more of these bone-destroying cells, which recruits more of the bone-producing cells. So there's more and more and more of these cells. There's a whole process of dysregulation that is what the tumor cell is going to be taking advantage of. Now, the important thing about this is that a lot of cancer models basically assume that you have a cancer cell that just grows in some sort of vacuum. But this vacuum is basically not a vacuum, it's actually the reality of this bone modeling unit. The cells that will survive, the phenotypes that characterize the tumor cells that actually thrive and proliferate faster than the competition, are the ones that can take advantage of the location, and that can take advantage of the interactions with the stromal cells, and can take advantage of basically the local microenvironment. These are things that people in the clinic don't take advantage of. These are things that we're trying to understand, because if we can disrupt the sort of benefits that tumor cells get from this stroma, we could potentially find new ways to treat this cancer. We have used this model to explore how these potential tumor populations are impacted differentially, because different tumor phenotypes would have different impacts by the different treatments you find in the clinic. And we have simulated a bunch of them. We have even simulated treatments that you don't find in the clinic to figure out whether important molecules like TGF-beta that actually have been incredibly well characterized in cancer but have not been put to clinical use could be leveraged through the use of mathematical models like this that can integrate data from both experiments, clinical data, and basically have this holistic view of the pleiotropy impact of this molecule on the cancer. So this is just one of the examples of things we're trying to do here. We're trying to capture these ecosystems so we can see exactly what drives tumors, in this case in the bone, so that we can actually stop them. Now, the bone is such an important organ that we can see models like this when applied for, basically, if a tumor is going to kill you in most cases because they metastasize. Prostate cancer, breast cancer, and lung cancer, which are the three main types of cancer you can find out there, metastasize preferentially to the bone. So trying to understand this context is going to be important not only for this particular problem, but for a lot of them. So without further ado, I'd like to obviously thank you guys for listening to this slightly shorter presentation than I expected, thanks to the lack of simulation movies. My group on the left, and Conor Lynch's group, which is the experimentalist who has been giving us the parameters for models as well as testing the hypotheses that come from the computational models. And I know it was mentioned before that people are looking for jobs. Coincidentally, I'm offering a postdoc position for someone who's interested in this kind of work. Thank you very much.